Good afternoon, everyone, and welcome to today's PHO Learning Exchange presentation on wastewater surveillance collaboration at York Region, Sampling Science and Solutions. My name is Ma'an Hasso, medical microbiologist at Public Health Ontario, and I have the pleasure to moderating today's session. Co-moderating with me today is Anna Majuri, clinical microbiologist at Public Health Ontario. I will now mention some housekeeping items. To ensure an engaging and optimal experience for all, the Zoom chat and audio-video features have been disabled for this session. Please feel free to submit any content-related questions in the Q&A function at the bottom of your window. They will be addressed during the discussion period. For technical issues, please email capacitybuilding@oahbp.ca. It is now my pleasure to introduce the speakers for today's presentation. Mark Penny, Supervisor, Environmental Monitoring and Enforcement, York Region Public Works. Karina B., Epidemiologist and Wastewater Project Manager, York Region Public Health. Vixie Fang, Data Scientist, York Region Public Health. Trevor Charles, Professor, University of Waterloo and co-founder, Metagenomic Bio Life Science. Elisa Everton, Lab and Wastewater Project Manager, University of Waterloo. Jen Knapp, Microbial Research Coordinator, University of Waterloo, and Michael Lynch, Vice President, Informatics, Metagenomic Bio Life Science. I will now hand it off to Mark to begin the presentation.
Thank you very much, Man. And hello, everyone. It's a pleasure to be here speaking about a subject that's close to my heart and bowels. Here at York Region, we are quite proud of the collaborations that we've built in order to derive surveillance value from wastewater. Next slide, please. First of all, just a quick disclaimer. This presentation was created by its authors and is an external organization of the PHO. PHO is not the owner of this content. Any application or use of the information in this document is the responsibility of the user. PHO assumes no liability resulting from such application or use. Next slide, please. Some of the learning objectives from today. One would be to describe the role of wastewater surveillance in evidence-informed public health at a local public health organization. Two, to recognize collaborative and sustainable infrastructure between public health, public works, academic institutions, and private labs. And three, summarize approaches to developing methodologies and technologies used in wastewater surveillance, sampling, processing, and innovative applications. Four, identify opportunities of expanding wastewater surveillance beyond COVID-19 to other priorities and disease targets. Next slide, please. So at York Region, we like to acknowledge that York Region is located on the traditional territory of many Indigenous peoples, including the Anishinaabe, the Haudenosaunee, the Huron-Wendat, and Métis peoples, and the treaty territories of the Haudenosaunee, the Mississaugas of the Credit First Nation, and Williams Treaties First Nations. Today, this area is home to many diverse Indigenous peoples, and we recognize their history, spirituality, culture, and stewardship of this land. We also acknowledge the Chippewas of Georgian Island First Nation as our closest First Nation community. Next slide, please. So, what is wastewater surveillance? It's a developing science. As back in the 90s in Europe, there was a few researchers doing it, but it really grew during the COVID pandemic and has become a vital tool in community surveillance. At York Region, we define wastewater surveillance as the ongoing monitoring of sewage for biological or chemical markers such as viruses, bacteria, pharmaceuticals, or illicit drugs to detect and track public health trends, outbreaks, or threats. Next slide, please. In today's agenda, we'd like to hit up a few things and talk about our important collaborations, our York Region wastewater infrastructure. It's important to know your infrastructure when planning wastewater surveillance. how public works and wastewater operations and public health have worked together, a timeline of recent events, program evaluations, some notable highlights, and some lessons learned. Next slide, please. I wanted to acknowledge that our wastewater surveillance program would not be possible without our relationships, especially the early support of public works. My colleagues in public works continue to provide in-kind support for sample collection and shipment logistics to ensure timely and high-quality data. We have also had support from academic partners at the University of Waterloo and the University of Ottawa. These scientists joined in the pandemic response, together through funding from the Ministry of Environment, Conservation, and Parks. As the landscape of wastewater surveillance continues to change in Ontario post-COVID, these labs have prioritized our public health surveillance objectives alongside their own research. We are fortunate to have successfully negotiated a continued partnership with Waterloo and their private lab Metagenom for the next year. Next slide, please. So it's important to understand your wastewater infrastructure as you embark on wastewater surveillance. York Region is an upper-tier municipality with 9 lower-tier municipalities. For wastewater, the lower-tier municipality is responsible for wastewater drainage pipes from the property lot line of homes and a series of small collection pipes, while York Region is responsible for the larger collection pipes, pumping stations, and for wastewater treatment. This may vary depending on the regional infrastructure or the regional setup. At York Region, our York-Durham sewage system is our largest system with over 120 kilometers of sewer pipes servicing the municipalities of Newmarket, Aurora, Richmond Hill, Markham, Stouffville, and Vaughan. This York-Durham sewage system services over 1 million people. It has 6 pumping stations, 2 large flow meter and odor control stations as well. Wastewater is treated at our Duffin Creek Water Pollution Control Plant, which is co-owned by Durham and York Region on Lake Ontario. The annual flow to Duffin Creek is greater than 130 million cubic meters. Travel time to Duffin Creek Water Pollution Control Plant on Lake Ontario is approximately 24 hours from our northern communities. The stars on the map are located in areas where we have sampled in the past for various pathogens. Most notably, our regular sampling for respiratory pathogens is from our Leslie pumping station at the blue star. York-Durham boundary meter at the red star is sampled for measles, while the yellow stars are locations we sampled for examination of antimicrobial resistant bacteria. And lastly, the green stars represent locations that we sampled this summer for H5N1 avian influenza. Next slide. So sometimes finding sampling locations is like finding the Goldilocks planet. It's hard to find a location that meets all of the criteria. Well, I think we're all familiar with the maintenance access holes seen on our roads on the picture on the top left, but many of us don't give them much thought. But they do provide access to our underground infrastructure, including our wastewater pipes. So though maintenance access holes are quite common, they really need to be located just right in order to be a good sampling location for wastewater surveillance program. They need to be easy and safe for staff to access, so not in the middle of the road. We want to be able to have staff easily access and be safe while they're doing so. The hole should not be too deep. We have to be able to reach the flow with a pole or with our 25-foot sampling hose. 3, we have to have good flow with no obstructions, because obstructions can block the flow and cause backup and age the wastewater, which can skew results. And 4, we have to be consistent and representative of our sampling objective. For instance, we wouldn't sample from a hospital drain if we were looking for a measure of community exposure. As seen in the pictures, we use a number of different sampling techniques based on the location being sampled. Sometimes we use a pole with a dipper to collect, and sometimes a peristaltic pump. The peristaltic pump is pictured in the middle and the upper right. Sometimes we sample right from a pump at our pumping station, which is the middle picture in the bottom row. The majority of our samples are a combination of 3 grab samples done 5 minutes apart, and then we combine those. And our samples are collected by our public works staff. Next slide, please. This slide shows some of our recent sampling that we've done. For COVID-19, influenza, and RSV, we're sampling 3 times weekly from October to May for the purposes of surveillance at our Leslie pumping station location. For measles, we're sampling 1 time weekly for emergency preparedness reasons from 2 of our sites, our boundary meter and our Leslie samplings, our sewage pumping station. For antimicrobial resistance, we did 2 times yearly from 7 different locations for a research project. We sampled from our hospital, 4 of our hospitals and long-term care centers. And lastly, in this summer, we sampled for H5N1 avian influenza from 5 different surface water that were frequented by birds. And now I'd like to pass on over to my colleague Karina to speak about the timeline of our wastewater surveillance program.
Thanks so much. Yes, you're right. I'd like to walk through a timeline of events because it's been a bit of an uncertain but very fruitful year for us. From March 2023 to July 2024, the MECP, or the Ministry of Environment, Conservation and Parks, did establish a temporary provincial wastewater surveillance program and provided in-kind funding in coordination of data across all Ontario public health units for COVID, RSV, and flu. And last year in June, MECP ended the provincial program. And at that time, York Region conducted an environmental scan across health units and various lab partners to gauge our funding opportunities. And though many health units did express interest, and several health units were able to continue wastewater surveillance with residual funding and resources, I think only Ottawa Public Health and York Region had secured funding over the last respiratory season. So in September of last year, York Region did sign a 1-year contract with Metagenom for wastewater surveillance, and subsequently, last October, we joined the Charles Lab at the University of Waterloo in a research collaboration through grant funding through NSERC and CBRF. And through these collaborations this past year, we've been able to expand our interest to other priority targets, such as measles, H5N1, and AMR, or antimicrobial resistance. So given the fruitful nature of this work so far, York Region has decided to extend our funding until 2027. Emerging from the pandemic, we had started to set some objectives with the intent to evaluate our own program. And at that time, it was helpful to determine whether the program could be cost effective and if the derived value of wastewater could really persist beyond COVID. So initially, we wanted to evaluate whether our 3 Tier A, B, and C sites might be reduced to a single site. And through quantitative analysis, we wanted to know which was our best site, and if this site could be reviewed across the 3 respiratory surveillance targets and prioritized for both early warning and concordance with clinical data. So once we had determined that sentinel sampling for York Region was feasible for surveillance, we decided to expand our evaluation plan for qualitative analysis. And in that phase, we aim to gain more insights into the attitudes of our own public health staff and leadership towards the perceived value of the program, and also to gain feedback of what could be improved. So for this evaluation, we were guided by the framework of the ECDC Handbook for Surveillance System Evaluation. We had chosen to evaluate these 14 parameters using the retrospective data over a 2-year period from 2021 to 2023. And using quantitative analysis, we reviewed data completeness, validity relative to clinical measures, sensitivity and specificity through cross-correlation studies, including for early warning lead time and timeliness based on the turnaround time of samples. And using our qualitative methods, we reviewed several other parameters through feedback from York Region Public Health Analytics staff, as well as from our senior leadership. And I'll turn it over to Vixie.
Thank you, Karina, for that overview. I'll talk a little bit about the quantitative and qualitative statistical methods that we conducted and the corresponding results. Firstly, in terms of quantitative, York Region conducted site-specific cross-correlation analyses on about 3 years of wastewater surveillance data that we've collected, and used clinical severity measures such as COVID-19 cases and COVID-19 hospitalizations, influenza percent positivity and influenza hospitalizations, and RSV percent positivity and RSV hospitalizations to see whether or not wastewater signals can provide us with early warning in terms of lead time. These statistical analysis was conducted on all of the sites that we collected data from. And out of all the sites, Leslie pumping site provided to be the best and most consistent site in terms of providing early warning. In terms of statistical results, we can see a relatively high correlation between Leslie sites and wastewater signals, such as disease cases and percent positivity and hospitalizations. We are seeing around 1 to 8 days in terms of Leslie providing us with leading early warning signals for COVID, 2 to 4 days for influenza, and 2 to 7 days for RSV. Next slide, please. Our program objective from this was to see whether or not we can make wastewater surveillance sensible and cost-effective for public health surveillance purposes. And we wanted to look at these two questions specifically. Are the disease signals, COVID-19, influenza, A, B, and RSV, across York Region sites consistent with the clinical data? And from our previous results, we can see that it was. The second was that whether or not sampling locations optimize wastewater signals for surveillance, and does it matter if this sampling site was upstream or downstream? And from the earlier results, we've seen that Leslie was the best and most stable early warning system for all targets, and Leslie provided us with unique upstream benefits in terms of all of the sampling sites. Next slide, please. In terms of qualitative analysis, what we have done pretty well with our program was that there was a very good awareness of this wastewater surveillance product and communications in our IDC team. And there's also no good agreement on the acceptability and usefulness of York Region Public Health Wastewater Surveillance Program. Additionally, we got a lot of significant interest to participate in research and surveillance opportunities after the fact. But there are also a lot of areas that we could have improved on upon the evaluation. Some were about the sustainability about the program. There were uncertainty regarding that area. And there's also a need for sharing technical knowledge in terms of how to interpret this, how to use this for additional support or data interpretation. Lastly, there is also an interest to include additional disease targets for representation. And as Karina and Mark has mentioned, we have kind of done that a little bit in terms of additional surveillance of Mpox, H5N1, and so on. Next slide, please. These were the specific results that we received from the evaluation analysis that we have conducted. One of the most significant questions was, what were the top 5 most important benefits of wastewater-based epidemiology? These questionnaires were issued to both leadership and staff, and we cleaned the data and looked at the results and conducted appropriate statistical analysis, such as Kruskal-Wallis, Fisher's exact test, and so on, to see what the top 5 important benefits are. As you can see here, there were a lot of consensus on the capacity emergency preparedness, early warning, disease detection, representativeness, and collaboration with academic partners for the big benefits. It's also very cost effective, credible, and in terms of privacy and confidentiality, because it's aggregated data, there are no violations there, which was also a very important benefit of wastewater-based epidemiology. Our statistical analysis indicated that there was a very marginal, significant difference between collaboration opportunities with academic partners between the responses from staff and leadership. Next slide, please. The second area that we wanted to look at in terms of evaluation purposes was to ask whether or not wastewater-based epidemiology has been used by said staff or leadership in the past 3 years. After conducting the survey, we have found that evidence-informed decision-making accounted for almost more than half of the usage of wastewater-based epidemiology. The next most used method of wastewater-based epidemiology was sharing with peers. and personal awareness. However, there are around 20 to 30% of staff and leadership that indicated they did not use any of this previously. And the only statistical significant difference that we have found between the results were that leadership tended to share with peers and staff tended to share with family and friends in terms of this. Next slide, please. Lastly, we wanted to evaluate the value of wastewater waste epidemiology in terms of proficiency, credibility, and applicability. And as we can see from this statistical graph here, nearly all staff and leadership rated wastewater-based epidemiology at least moderately credible and applicable. So that was around 100% credibility and 80% applicability. However, only half, which is around 47%, consider themselves moderately knowledgeable in wastewater-based epidemiology. So this identified areas where we could have improved on in the future as well. Next slide, please. After identifying that, we wanted to see what aspects of the current program can be improved and what can we extend to. So in terms of the responses, we have seen a lot of interest in knowledge sharing, additional disease targets, which I'm happy to say that we have achieved, data accessibility, collaboration, long-range planning, and sampling approach. Next slide. This was the poster that we have presented on of our qualitative and quantitative analysis for TOPAC. And I won't go through all of this with you guys, but I just wanted to kind of draw your attention to our conclusions. The quantitative conclusion was that our Leslie site was the most reliable and consistent location for monitoring disease targets, and we found a very strong association between the wastewater signals and clinical indicators. The qualitative analysis told us that there was a very good awareness for wastewater surveillance products and communications. There was consensus on the agreement of acceptability and credibility of the wastewater surveillance program, and there is also strong interest to participate in research, collaboration, or additional surveillance opportunities in the future. Next slide. Sorry, go ahead.
So since last year, we've continued to maintain this program in setting new objectives. First, we continue to support our lab partners in the development of assays and diagnostic extraction methods, and through the in-kind contribution of water samples. We'll also continue wastewater surveillance for the respiratory season on our 3 targets. And through the CBRF grant too, we're still collaborating on antimicrobial resistance research. And with an eye on expanding testing to other disease targets, this year we've been able to be supported by the Charles Lab in surge monitoring for measles and surface sampling of H5N1. And finally, with all surveillance, we hope to repeat our evaluation again for new disease targets, as well as to learn more from our new sampling strategies too. So I'm really proud to share that our team has had many notable highlights with wastewater surveillance over the most recent years. So not only were we one of the early adopters to establish this program in September 2020, but we were also first to democratize COVID data and share it in near real time through our external facing dashboards. And through the Omicron wave, when testing of the general population was overwhelmed, we leveraged wastewater signals for predictive modeling to compare the impact of different pandemic control measures. We've also been fortunate to be collaborators in 2 multi-year NSERC Alliance grants, one with the Servos Lab at the University of Waterloo and with the Delatola Lab at the University of Ottawa. And as a result, our pandemic pilot program was recognized in 2023 by the Ontario Public Works Association for our innovative program. And we've also shared some of these early experiences through research publications, as well as a case studies book chapter. And finally, you might have seen our team at Topic this year presenting some of our evaluation results as well, as we continue with our knowledge exchange with our peers. And finally, again, we're very proud to say that we have this program in place until 2027. I wanted to wrap up my portion of this presentation by presenting a couple of helpful takeaways that were useful for us when we were considering a sustainable program. So first is owning wastewater-based surveillance as a sustainable tool. Wastewater surveillance is a useful tool, but as we know, not all surveillance systems survive the test of time. And thankfully, prominent agencies like the US CDC or the European Commission have already mandated wastewater surveillance to be a continued investment. But to prove its value, especially as a regional program, we needed to move beyond just it being a pilot program for COVID, but more importantly, to show cost effectiveness and that value add. So through conducting these validation studies of our data across all our sampling locations, and also going to market to understand cost models and potential research collaboration opportunities, we were better able to streamline this program. In this process, we also were really intentional about understanding our wastewater infrastructure and how it affects data quality, and really focusing on this application for disease priorities. Secondly, target selection is a hot topic, but the prioritization of disease targets varies widely across our health units. We've heard from some health units that they'd like to prioritize opioid surveillance, while others are prioritizing vaccine preventable disease and more so STBBIs. And it's definitely not one size fits all. And we've really had to think critically about our own York Region needs. But where we have unreliable data for disease burden, there's always these opportunities to leverage wastewater for these insights. One of the challenges of wastewater surveillance is that we often validate a new surveillance system against clinical standards. And in some cases, wastewater indicators might be superior to clinical indicators as a more representative and timely metric of community disease burden. So it's important for epidemiologists to develop a deep understanding of our infectious disease indicators, including how they differ, their strengths and weaknesses, and also to use wastewater data together with other surveillance indicators to provide a more accurate picture of risk. Moving on to investing in academic and diagnostic partnerships. We can't emphasize enough that a program like ours stands on the shoulders of our academic partners and trailblazers that contributed their expertise and resources during the pandemic response. So we really appreciate the amount of work that it has taken to develop all the extraction methods and assays, quality control processes, and logistics to make even this kind of surveillance possible. So it's really important for us to continue to nurture that dialogue, the cross-pollination of skills, and also to lean into the new opportunities where we might collaborate. Further to that, in a new and fast-paced field such as this, it's important for our team to continuously assess whether the juice is worth the squeeze and whether it aligns with our values and core business needs. Internally, our infectious disease team not only partners with Public Works, but also with Health Protection Division, as well as the Digital Clients Experience branch. And these collaborations really ensure that we can continue to navigate this new science in the most effective manner. And finally, building surge capacity for emergency preparedness. Wastewater as a surveillance tool has really faced its share of challenges, especially when it came to data interpretation for lay audiences. Much of the knowledge that we gained with setting up the surveillance system, from partnerships to processes, all of this should be thoroughly understood, and this knowledge should be safeguarded. So for wastewater surveillance, this year it has undeniably added to our toolbox of tricks. And at York Region, we found it helpful to reach for this tool several times in the past couple of months, whether it was for monitoring of the respiratory season or for determining risk of our own local measles outbreak. If we don't use it, we might lose it, and there's no telling when it might come handy next. So in that way, we feel like keeping polished on this process has been really great value for our search capacity. With that, I will turn it over to the Charles Lab, to Dr. Tretbow Charles.
Thank you, Karina. Our lab at the University of Waterloo was involved in the Ontario Wastewater Surveillance Initiative during the pandemic. We specialized in variant detection using sequencing. This set us up nicely to open up new research initiatives using wastewater, some of which we'll tell you about today. The continued partnership with public health units such as York Region has been crucial to this work, and it fits well with the ongoing surveillance services provided in partnership with Metagenom Bio Life Science. So today, we're going to report on our work in discovering novel antimicrobial resistance, novel viruses, and also detection of H5N1 in environmental samples. We'll also tell you about the work we are doing in developing and refining assays for flu, RSV, and measles. Next slide, please. Next slide, please. Get there, thank you. Great. So this work has been supported through a number of funding sources, without which we would not be able to do any of it. And so we'd like to acknowledge it here. Current funding is an NSERC Alliance grant for antimicrobial resistance work, and CBRF Inspire funding for the pathogen surveillance work. I'd like to also acknowledge our key collaborator, Professor Andrew Doxey. Next, I'll introduce you to Jen Knapp, who will tell you about some of the research. Thank you.
Thanks, Trevor. So the broad goals of our antimicrobial resistance, or AMR research, are to first get an idea of which AMR genes are usually present in these types of samples and what levels might be typical for our area, and then to see if we can discover some novel AMR genes or host species. There are a few approaches that we take in parallel. So starting at the top left of this diagram, the first thing we do is extract all the DNA from the wastewater sample and sequence it. So that's going diagonally down towards the center of the diagram. This tells us broadly which organisms and AMR genes are present and their relative abundance. We can use this information to design assays that target specific AMR genes or classes of genes to sequence more deeply and specifically. However, this type of analysis is restricted to telling us about previously known and described AMR genes, since this data is compared to various databases for analysis. Another thing we can do is directly plate this wastewater onto agar plates containing antibiotics of interest, and then culture the resistant microbes. So this process is pictured on the left side of the diagram going downwards. DNA from these colonies is then sequenced to determine their species and if they contain a known AMR gene. This is valuable, but we know that only a small fraction of microorganisms are culturable in the lab, so we'd be missing a lot of information. So the way we bridge the gap between these two methods is what we call a functional metagenomics approach. This is pictured along the top, towards the right, and then down the right side of the diagram. This is where we take the DNA that was extracted from the wastewater sample and break it into larger fragments. We then ligate these fragments into a broad host range cosmid backbone and transduce this library into a host organism, in this case E. coli, but we have also made libraries in Pseudomonas putida and are working on several other host organisms, including Bacillus subtilis. Then we plate this library onto antibiotics of interest. We started with ampicillin, meropenem, imipenem, and kanamycin, and are still working on assays for vancomycin, astronam, and colistin sulfate. Colonies that pop up on these plates are then carrying a cosmid with a wastewater DNA fragment that is able to confer that antibiotic resistance to the host, which we then sequence for characterization. We expect to identify known AMR genes through this method, but there is the potential to discover novel AMR genes here as well. If the insert sequence does not map to any known AMR gene, but is conferring resistance to a specific antibiotic, further experiments can be done to narrow down which part of the insert is responsible for this resistance. Another advantage to this approach is that we can examine the rest of the sequence in the insert and get information about which species it came from. Next slide, please. So here are some quick examples of the kinds of results we can get from these experiments. On the left-hand side is a graph of all the different drug classes of AMR genes that were found in 4 different wastewater samples just through sequencing. For our interest in carbapenems like meropenem and imipenem, for example, we'd be looking for genes that fall into the beta-lactam resistance group, which is the second most prevalent group in these samples. For the metagenomic libraries, I'm showcasing here on the right-hand side that 8 libraries that we constructed and hosted in E. coli. We screened them on imipenem and meropenem, and a total of 30 resistant clones were investigated, most of which had easily identifiable AMR genes present, but we did find one interesting clone without any AMR genes when screened against 5 representative databases. There could be a novel AMR gene here, so we're investigating further. So far, we know it's from an Undibacterium species, and we have several candidate genes that perform similar functions to other AMR genes that we will test both bioinformatically and experimentally to see if any of them confer the resistance. Next slide, please. Here I'd like to highlight some work by Julie, a postdoc in our group. She's been working with the organisms that were isolated after plating wastewater directly onto plates containing imipenem and meropenem. She started with 56 isolates, 33 of which were Pseudomonas species, so her initial focus is on these. After sequencing the genomes of these Pseudomonas isolates, she determined the AMR genes that were present, and as you can see from this graph, there are quite a few, but many of them did not have identifiable beta-lactamases or carbapenemases that would explain their resistance to imipenem and meropenem. She picked 4 representative species, denoted with the stars, and made metagenomic libraries out of their genomes to screen on imipenem and meropenem to narrow down where the resistance is coming from. This work is currently underway, and she has 12 resistant clones that she is investigating from these screens. Next slide, please. In the same theme of new discoveries, Shahrzad, one of our PhD students, is hoping to discover some novel viruses in wastewater. So when we sequence all the RNA in a wastewater sample, only about 1% of the data is from viruses, while the vast majority is from bacteria. Most of the viruses we find in wastewater are plant viruses, which are from the plants and plant products we consume, and phage, which are viruses that infect bacteria. And only about 1% of that initial viral 1% are sequences from human infecting viruses, and even less of these have pandemic potential. We would love to have an untargeted approach to detecting these human pathogenic viruses without the need for assay development in an emergency situation. So the challenge is to increase the resolution to enrich for viral sequences, which we can do through both experimental methods like filtration and centrifugation, and also through bioinformatic methods. Shahrzad is working on these viral enrichment strategies, but is also developing a strategy to detect potentially novel viruses in these wastewater samples. Typically, after sequencing, the reads are assembled into longer stretches called contigs, and then they're fed into a tool like Kraken-2, which maps them to a taxonomy database to tell us what's in there and how much of it is viral. RNA viruses all contain a conserved RNA-dependent RNA polymerase, or RDRP for short, which is necessary for the virus to replicate its genome and therefore itself. Shahrzad examined all of her contigs that contain an RDRP sequence and noticed that some of them were not classified as viral sequences by Kraken-2. But since viruses are the only possible source for these sequences, she investigated further. The table on the right shows some of the top hits for RDRPs that were not classified as viral by Kraken-2, but when run through BLAST, their closest match is some kind of viral sequence. You can see here in the identity column that they each only have around 70% identity to their closest match, meaning that we don't know exactly what these sequences are, but we'd love to find out more and see if we can recover more of their genomes for further research. I will point out too that these are very unlikely to be human infecting viruses, as those will have been much better categorized through clinical sampling. As you can see, none of these closely related hits in this table are human pathogenic viruses. Most of these are associated with fungi or plants. The graph in the bottom right shows how many of these RDRP containing contigs can only be identified down to the phylum or family level, but not to the genus or species level, indicating that there are potentially lots of novel viruses for us to find and characterize. Even if they are not completely relevant to human health, The methods developed through this work will serve as one building block in a suite of tools to keep us apprised of what viruses are present in our wastewater. Next slide, please. There is one animal virus that we've been watching pretty closely, and that is avian influenza, or H5N1, since there have been many outbreaks in wild and commercial flocks of birds and recent spillover into dairy cows in the US. We're developing and testing assays to detect H5N1 that can be used in surface water samples as well as wastewater samples so that we can be ready in case this virus needs to be monitored more closely. York Region's Health Protection Team facilitated sampling at 5 lakes or ponds across the region, from both the surface of the ponds and from the sediment at the bottom, giving us a total of 10 samples. The total RNA was extracted and tested with an HA segment PCR with H5 specific primers, so only samples with H5 in them would be positive. We found H5 PCR positivity in 2 sediment samples denoted with stars on the map. We sequenced these, and blast analysis showed that the closest similarity was to H5N1 and related H5N6 and H5N8 isolates. When we place it on a phylogenetic tree, our sample is in red on the tree at the bottom. It clusters with 2 samples of H5N1 in geese from the database, meaning that these are likely true positives from wild birds. Sharzad's next step with this work is to design primers that will amplify more of the H5N1 genome so that we can have more information about clade-specific mutations to learn more about H5N1 presence in these environmental samples. I'll turn it over now to my colleague Alyssa to talk about some more targeted assays that we've developed for use in wastewater.
Thank you, Jen. On the targeted surveillance side of our expanded wastewater work, I would like to showcase 2 tiled amplicon sequencing methods developed by PhD candidate Delaney Nash. These target one flu strain, H3N2, and one RSV strain, A. These were both chosen because they were the most prevalent circulating strains of each during the study period. You might be familiar with tiled amplicon sequencing from the Arctic assay that was used during the SARS-CoV-2 pandemic. The RNA in wastewater is usually very fragmented, so long PCRs would not be successful. Tiled amplicon assays are an effective strategy for whole genome sequencing of target viruses in wastewater because the primers are designed to amplify short fragments, which we call amplicons. These cover the entire target genome in a staggered and overlapping fashion. This is shown in the illustration on the upper right of the slide. After sequencing, these amplicons are reassembled by mapping against a reference genome, resulting in nearly complete genome coverage which captures the diverse mutations in the wastewater sample caused by circulation of multiple strains. On this slide, flu H3N2 is presented in blue and RSVA is always represented in pink. In the case of H3N2, the percent coverage of the genomes, which is shown as trend lines on the plots to the right, correlate well with the percent positive cases in York Region, which are shown as bars. Peaks in the percent coverage even appear a couple of weeks ahead of cases and spikes for RSVA. The mutation frequencies over the genome can also be deconvoluted using variant calling tools. The bottom left of the screen shows the variants predicted to explain the mutation diversity by our in-house variant calling tool, alvoc. On each of the 4 plots, the subtypes of H3N2 or RSVA are on the y-axis, and the sampling date is on the x-axis. The heat map percentages represent the percent of each sample made up of each subtype. This type of assay can be developed for other targets with whole genome sequences available, and many have been published. The alvoc tool can be adjusted for any target with a next strain tree, which can also be made by researchers, so this gives us great flexibility for future targets of interest. Next slide, please. Okay, next I would like to present our work so far on measles detection. During the recent Ontario outbreak, we adopted 2 CDC qPCR assays, which were designed for clinical testing, and optimized our workflow to make use of them on wastewater samples. The first, which we call measles all, captures any human measles variants, including the vaccine strain. If we receive a detection for this measles all assay, we then test with a second assay, which only detects the vaccine strain. If we don't get a detection for the vaccine strain, then the all measles signal must have been from wild type. But if we do get a vaccine detection, we know that at least some of that all measles signal can be explained by vaccination. On this plot, we see the copies per mil of raw wastewater are plotted on a log scale over time so that we can see our low copy detections. Data points from the measles all assay are represented as circles, and data from the vaccine assay is marked as squares. The two colors differentiate the two sites that were sampled in York Region. The clinical cases are plotted on a second y-axis and shown as bars. Later on in the most recent group of clinical cases, we had multiple samples which showed clear detection, but at lower copy number than our limit of quantification, or LOQ, which is shown as a dotted line on the plot. These we know are detections, but are not confident in the quantity. York Region was still able to use this decreasing signal as an indication that additional transmission outside of their clinical records was not likely occurring at this time. A single amplicon sequencing assay capable of differentiating the subtype is also in use in our lab, but from pilot samples, it appears to have a higher limit of detection than the signal we saw in this study. That concludes what the Charles Lab Research Group is sharing today, so I will now pass the presentation on to Michael Lynch from Metagenom Bio. Thanks.
Thank you very much. I'll just introduce and talk about our wastewater pathogen testing and our translational research. Next slide, please. So testing at Metagenom Bio, we have a flexible and innovative testing strategy with rapid test development. And this is really important for emerging and evolving targets. Our fast lead time with integration between academic research and industry government is a strength where we focus on translational knowledge. And this rapid development and deployment of these particularly sequencing and amplicon assays like qPCR and tiled sequencing. We in our group have extensive experience with high throughput sequencing, qPCR, and analytics. And one strength of using a sequencing approach is searching existing data that has been generated historically for new queries which can study trends predating an assay. We have a wide variety of clients in agriculture, government, industry, engineering, and mining. Next slide, please. Two major standard public health assays we have available are the respiratory virus panel, which covers SARS-CoV-2, influenza A and B, RSV A and B. And then there's also tiled sequencing and variant detection available for many of these. We also have a multiplex bacterial assay that covers E. coli O157H7, Salmonella species, and Listeria. Next slide, please. We also have many target-specific amplicon assays covering bacteria, fungi, general eukaryotes. Some of those two are of public health interest, Cryptosporidium and Giardia species, along with many plant pathogens that we apply in an agricultural or hydroponic context. And we also have the strength of rapid assay development. Next slide, please. I'd like to just briefly discuss three examples where our expertise and rapid development solved or had a unique approach to three problems in industry. First, antimicrobial resistance. Next slide, please. We were able to use target selection guided by analytics. So we do universal sequencing of a gene that works like a barcode, and that guides any analytics and future qPCR to be more effective and more efficient. And this prediction of gene presence based on this microbial community composition was confirmed by qPCR. So allowing us to cast a very wide net to inform analytics and hypothesis generation. Next slide, please. Second example is plant virus hunting by RNA sequencing. Next slide. We had a client with severe symptoms in bok choy that looked a lot like Pythium, standard Pythium symptoms, a common plant pathogen. But the cause could not be identified by other service providers and any assays for Pythium. So we did a universal sequencing, no major plant pathogens, but the samples were dominated by Opidium, which is a vector for plant viruses. So our next approach was a virus enrichment and RNA sequencing, doing a wide RNA sequencing for all targets. The result, we identified a campestris chrysovirus that infects cultivars of Brassica rapa like bok choy that has symptoms consistent with grower experience, and that would not have been observed in any other standard assay. Next slide, please. The third example is Fusarium wilt in peppers. Next slide, please. This is a devastating disease, common plant pathogen in hydroponic systems, which causes a wide variety of symptoms. The most important of these is vascular wilt. And it is a devastating disease in Ontario greenhouses, significantly emerging since 2021. And this work is in collaboration with OMAFA and AAFC. Next slide, please. We observed that, yes, Fusarium was common in almost all severe cases of disease, but also co-infections with severe pathogens like Pythium, and there were compositional subgroups that impacted how significant wilt was in these samples. Next slide, please. So the strength we have is this flexible price and sample flexibility, no need to commit to contracts. We can process as many or as few samples or targets as needed. And we work closely with health units to develop effective monitoring strategies, target sites, and frequencies. Thank you very much.
Thank you to all of our speakers for that insightful presentation. We will now move into the Q&A session to answer questions from our live audience. If I could please ask that all our speakers and our co-moderator, Anna, join us at this time. So maybe I can start the first questions, and I think this one will be directed to your public health colleagues. So the questions state that, do you have evidence that the public changes their behavior based on the dashboard data?
I'll answer this in little parts. So based on our integrated respiratory disease dashboard, we do present data on wastewater for COVID, RSV, and flu separately, but all together on the dashboard. We do know that we have good usership of this dashboard, although the dashboard is tailored for frontline workers and healthcare providers too. We have received good feedback from the public that they'd like to continue to see wastewater data for their personal use, especially with those that might be immunocompromised or might be frontline workers that need to care for the elderly as well. Within those risk groups, it's really important for them to have their own data to exercise risk mitigation. Otherwise, we routinely use this data with our consultations with our hospital partners and long-term care partners, and that is used on a routine, regular basis. And that is more for surge planning in case of seasonality or a high capacity within the frontline staff.
Okay, thank you. So maybe there's another question also maybe will be addressed to your colleagues. Did you consider expanding the wastewater surveillance program to mpox?
Not at this time. For us at York Region, we have very few mpox cases. We have had under 20 cases in the past 4 years. So at this time, it's not a priority. However, we do prioritize other sexually transmitted and bloodborne diseases as a group, as a whole.
Okay, thank you. There's one question maybe to the Waterloo University colleagues. Are there any commercial kits to enrich the sample's viral sequence data?
Sure, I can answer that. So that's a really good question. So there are commercial kits that allow you to enrich, but the challenge with those is that they are based on known sequences for viruses. And really what we would like to be able to do is to identify viruses that are completely novel. So this would in effect screen out that novelty. And so that's basically why we're staying away from using those commercial kits.
Thank you. Great. And maybe one question also, I'll switch to the measles, also to the Waterloo University colleagues. Have you tried multiplexing like the measles assay with like combining both like the vaccine strain targets with the measles viral targets so that you can detect them both in one shot instead of doing like a screening? Then if it comes positive, you go with the vaccine strain?
Yeah, for sure. We have tried when we get new targets for qPCR that we want to multiplex, we have to make sure that nothing looks funny when we start multiplexing them. And in this case, while it should very well be possible, our probe for the vaccine target has caused issues in the past.
Okay, thank you. There's one question specifically to STBBI are being explored, monitored in wastewaters like STIs, bloodborne infections.
Sorry, could we clarify on that question if that's in general or at York Region?
Maybe in general, maybe I'll consider it with maybe Waterloo University. I know you've been trying like some other viruses, but have you tried like an STBBI targets, like bacterial or viral?
We haven't done any STBBI monitoring in our lab, no.
Okay, thank you. Okay, and maybe the last question, we have like 1 minute more. Maybe this is for York. Do you know if you York Region wastewater surveillance methodology is comparable to the PHAC wastewater surveillance programs?
Perhaps Waterloo might speak to the protocols in more detail, but we do align our protocol with the US CDC during the pandemic. Our infectious disease trends, the sampling is collected 3 times within a 7-day period to ensure that monitoring is reliable. And we do adhere to the PHAC sampling strategies as well.
Okay, thank you. And maybe this is the last question I would say to your colleagues. What do you do differently if you find a measles vaccine strain versus a measles wild type? Are there any different public health actions?
At this time, we haven't had too many detections to work off of, so we're still assessing as the data comes in, but we are working with our colleagues at the Public Health Agency of Canada and Public Health Ontario with the results.
Okay, thank you. Okay, so as we wrap up today, PHO learning exchange session, I would like to thank our speakers, Mark Bainey, Karina B, Vixie Fang, Trevor Charles, Elisa Everton, Jen Knapp, and Michael Lynch for presenting. And I would also like to thank our co-moderator, Anna Majuri. Finally, I would like to thank everyone who joined us for today's event. You can expect to receive a brief and anonymous survey for today's session. Please try to complete this to help us improve our programming. Thank you and have a wonderful day.